that comment. So look, it's a great pleasure to welcome Dr. Ben Herbert to the Biobank today to present for us. I think some of the people who are joining us from Sydney will know of Ben. He has many years' experience in the fields of protein chemistry. His expertise is also in sample preparation, fractionation and proteomics. He has gained international recognition as a key innovator in sample preparation, fractionation and the use of proteomics in research. And some of you will know that he has co-founded a number of companies, including Proteome Systems and his current company, Sangui Bio, where he's the Chief Scientific Officer. So he has mainly worked in technology development and commercialization, and he has a lot of experience in securing investment for companies in the biotech and healthcare industry. And he has a track record of creating, protecting and commercializing intellectual property. So I think that's quite unique experience and probably very different from many of the people that are joining today from either the biobanking sector or the research sector, but something that we need to hear a lot about. So I was very excited to invite Ben, particularly to talk about microsampling technologies, because I think something that we don't talk enough about in biobanking is, in addition to consideration of financial sustainability, we need to really pay a lot more attention to the environmental sustainability of biobanking, and that also helps with our financial bottom line. So it's a great pleasure to welcome Ben today and to hear about his talk, which is titled Doing More with Less, Microsampling Technology Enabling Novel Biomarker Discovery. Thanks a lot again, Ben.
Thanks, Jenny. Thank you for the invitation, and thanks to everybody who's online to listen. So I thought as we go along here, now I've got to figure out, there we go. So when we think about blood, so this whole talk will really be about blood marker analysis, but as we go, you'll see that the kinds of things we're doing would be applicable to other sorts of samples where you've got cells suspended in fluid, even things like tissue culture. But I'll talk effectively exclusively here about blood. And when you think about blood as a collection of different cell types in liquid, there's a whole lot of ways that we could have got to where we are in terms of blood marker analysis. So I thought I would just spend a little bit of time talking about, how did we get here with blood marker analyses? And I think there's really the three factors that I'll talk about here are clinical chemistry, dogma, and proteomics. And if we go back in time a little bit and look at clinical chemistry, really the thing that drove the real testing of serum and plasma is effectively hemolysis and the colour and absorbance of blood. And when you go back and look in the literature, what you'll find is that if you look at that graph on the right hand side there, of course hemoglobin absorbs very strongly. So if you're doing an absorbance assay and you've got a hemolyzed sample and you've got any analytes you're interested in that wavelength area, it's a total disaster, and there's a large number of papers that talk about that. And of course, if you're doing colorimetric assays, which were very common, then the red colour interferes with those colorimetric assays as well. So there's a lot of that historical work that's really driven where we are today, and the focus on working really with serum and plasma because they get around that problem. The next slide here I just thought was interesting. Occasionally you run across people that have perfect names, and this gentleman here, whose last name is Blank, working on a study trying to understand how to improve assays, I just thought he had the perfect name, so that's the only reason that's there. The second aspect of this is dogma, and what I mean here is the central dogma of molecular biology. And here are the four people that really contributed to that. Only three of them won a Nobel Prize, so I'll leave you to guess who didn't win. The critical aspect of this, of course, was that everything that didn't fit within that central dogma of molecular biology was really left out in some ways, hence the crying red blood cell here on the side, because they don't fit. And so they've largely been ignored for quite a long time now, even though red blood cells make up the majority of cells in our body. And if you could take all of those 25 trillion red cells in a person and lay them out flat, the surface area would be about 3,300 square meters. So they have this enormously large area for interaction. And when you think about blood, there's 1,000 red blood cells for every other cell in the blood. So there's a lot of interaction going on. And really, I think we're only just catching up with the kind of things that red blood cells can do. And that's really where the Sangui Bio story started, was we were looking at cytokines, and like everybody else, we were looking in plasma and serum at cytokines, and we, like everybody else, were ignoring all of the cellular component of the blood and throwing it away. And the problem that we ran into, and we were just lucky, like a lot of things that you find, we happened to be looking at MIF, which is macrophage migration inhibitory factor. And this work was being done by Elizabeth Carston, who was a PhD student at that time and went on to co-found Sangui Bio with me and Alan Little. And her PhD completely changed at this point when we were struggling to get sensible, reproducible answers looking at MIF in serum and plasma. And we just did some experiments where we froze whole blood. And when we did that, we got these answers that there was 30 micrograms per mil of MIF in whole blood. And Liz went to the flow cytometer and purified the different cell types. And we found that 99% of that MIF was in red blood cells. Wow. So that's an interesting example in its own right of everybody, or every protein, every cell type doesn't really just get to do one job. So MIF is an inflammatory chemokine, but it's also an oxidoreductase. So there's reasons why you would expect large amounts of MIF to be in red blood cells. And we weren't the first group to find MIF in red cells, but that had been sort of lost in the literature. And that really led us on to looking at red cells and saying what other cytokines might be there. So we've published a range of papers now looking at what cytokines you find apart from MIF. And in our case, when we first did this work, we were looking at 48 different cytokines, and almost all of them were present in or on red blood cells in a dynamic way. They bind and they release. Red cells interact with white blood cells. They change their profile when they're exposed to cancer cells. And so we've done a range of work in that kind of area. If you look at the paper in the top right, there's a table or two in there where Liz has broken down the plasma concentration as well as the corrected red cell concentration. She's got a really beautiful method for purifying red cells. So what we found was that in many cases, red cells had very high levels of cytokines compared to what you saw in plasma or serum. And so we developed a method for fractionating blood. One of the great things about red blood cells is they're so heavy that you can get them out reasonably pure quite easily. And even from frozen blood, you can remove red blood cell membranes. We've discovered that proteases play a key role in that binding and release, and you can perturb what happens to red cells by using protease inhibitors or cocktails of inhibitors. So for quite a long time, we worked this way looking at blood. We've done studies where we've looked at liquid blood in diseases like this one here as non-small cell lung cancer. And we found cytokines that are significantly different in both the plasma and in the red cell component, looking at red cell lysate as well as looking at red cell membranes. So this in its own right was interesting, but what we ran into frequently was when we wanted to scale up and do more than sort of 10, 20, 30 samples, it was difficult to do that. And of course, it's expensive to fractionate blood and start looking at all of these different fractions. And a lot of groups that we talked to pushed back against that and said, you know, this is too slow and it's too expensive. So it became quite clear to us that scale up really required a different approach, and that while liquid blood would remain, I think, the gold standard because of the amount of material you can get and the types of fractionation and the analyses you can do, that dried blood spot or microsampling enabled a whole range of things that were not going to be able to be done with liquid blood. It would be much easier to get samples from people at home and to get them much more frequently. It was a lot cheaper. You didn't need as much material, so you could sample people more frequently, and it wasn't a problem from an ethics perspective. We really settled on what I've called here VAMs, so that's the kind of technical term for this. The device that you see there is made by a company called Neoterix, who are based in Los Angeles, and they were recently bought by Trajan, who is an Australian company based in Melbourne. And the plastic tips that you see there are porous polyethylene, so they're not paper, and that turns out to be a real advantage, not just because it's very precise in the amount of volume that it absorbs, but because that porous plastic material doesn't disintegrate like the old-fashioned paper does for dried blood spots. So you can wash that and extract it multiple times, and you'll see why that's important as we go along. So this was one of the first studies that we did looking at people and sampling at home at multiple time points, and these are a whole range of cytokines as well as CRP, and it's all participants at all time points, and the data are normalized. And so what this sort of thing enables you to do is to really quickly see who's different from everybody else. So in this case, looking at a score that incorporates BMI and lifestyle, so drinking, smoking, diet, exercise, and we rapidly saw that there were correlations between people who had the worst lifestyle and people who had significantly different markers, in this particular case, leptin. And so the thing for us at this point was we had a platform where we could do liquid blood when people were in clinic, and we could supplement that with dried blood spots when they were at home. But the problem with that really is that from a sampling perspective, we've got a quite good solution. We're able to look at inflammatory markers like cytokines, but in terms of everything else, we're doing at this point what everybody else is doing and ignoring all of the other components in blood, mostly because it's such a technically difficult problem that we were ignoring it for that particular reason. But what the market really demands, and when I say the market, I don't just mean the commercial market, the research market as well, is really looking for biomarker levels. Every group that we deal with in one way or another wants what is here at level 1. So if they're working on heart failure, they have specific heart failure markers they want to look at, and they really like them to work both from liquid blood and from dried blood spots. They are interested in inflammatory monitoring, so they're interested in things like cytokines, chemokines. Most of them are interested in the role of things like red blood cells and non-conventional ways of looking at it. But all of them, from the largest pharmaceutical companies that we deal with to individual research groups, also want to be able to do discovery level work, particularly things like proteomics, and of course the core technology now is mass spectrometry. And they would frequently ask, can you do these things from dried blood spots as well as from the conventional venous blood sampling and looking at serum or plasma. The other things, like we've done SNP array work from dried blood spots, and that works quite well. There's plenty of DNA in there to do SNP arrays from. We've used 30 microliter Neoterix devices. We're dabbling a little bit in blood typing. We haven't really done any work in metabolomics, but I know other groups have from dried blood spots quite successfully. So the reason that we've not really done anything in that other sort of area, the discovery area, is really this issue of dynamic range. And what I mean there is the concentration range from things like albumin and hemoglobin down to things like cytokines is so large, and blood, particularly plasma and serum, are so dominated by this small number of 20 or 30 proteins that it's a very difficult sample to work on. And in an unfractionated way or undepleted way, what you tend to find is you just see albumin and immunoglobulins and things like that in the mass spectrometer. And the examples of that really, this is just one paper that's a good example. This was published in 2019, and they only looked at 11 samples, 6 Alzheimer's disease and 5 controls. And the reason they did that was that they created 180 fractions in chromatography fractionation, and then they analysed each one of them. And they said this represents one of the deepest serum proteome analyses, and they're correct. The problem is each sample took 60 hours to do. So this is, that's why you can only it took them 1 month to do 11 samples. And prior to this, they'd actually published another paper in one of the cell journals with Matthias Mann, who's one of the real legends in mass spectrometry proteomics. And they said, look, we've got this really short way of doing this, and we can identify 347 proteins, but it only takes sort of 2.5 hours of sample prep and analysis time to do that. The problem is that's only 8% of the number of proteins they identified when they did their 60-hour method. And then they said, look, there's a 16-hour version of this, and that then takes 27% of the time, but they're only getting 23% of the proteins. And so I thought, I wonder what this really looks like on a graph. So here's those 2 papers graphed out. And I've now gone and looked at a whole range of other papers. So the blue line and the blue dots on that line are a whole range of papers on plasma proteomics by LC fractionation and then LC MSMS. And there's an incredibly good correlation between the number of identifications and the amount of time it takes. The 3 dots with the red circles are all PBMCs. And so they clearly step away from the line, and PBMCs, of course, are relatively easy to get from whole blood, and they don't suffer from the dynamic range problems that you've got in serum and plasma. So you can see how effective that is. And so we were really thinking, what could we do in this kind of space? And it's a very crowded space. There's a lot of people in here. And we started talking, it was really led initially by Mark Malloy, who's a colleague of mine way back from our PhD days. And Mark is the Professor of Colorectal Cancer Research now at the University of Sydney, and he's based at the Kolling. So we started talking about doing mass spectrometry proteomics from these dried blood spot devices. So when you think about what these look like, when you put whole blood into them, you've effectively got this plastic sponge, and whole blood is going in and everything becomes trapped inside there. And what you've really done at this point is you've solidified blood. You've turned it from a liquid material into a solid material. And you can now extract that. And what you're then left with is the membranes and the DNA and everything of those cells are trapped inside this device. They don't come out. And we discovered that by accident because one of the guys in the lab was trying to reuse tips to just do method development on, and we could never get them empty and reuse them. So we realized that what was stuck in there were all of the membranes. So what we've now got is a series of methods where we can do sequential extraction on these tips. We can do a very small volume extraction and do multiplex immunoassay and look at things like cytokines, and then go back and wash the tips without fear that we're going to wash all the membranes out, and then in situ digest them with trypsin and take that set of peptides to the mass spectrometer. And so we published a paper this year with Mark and his group showing what you could do with this, and we've looked at a range of different samples and sample prep and washing technologies in this particular paper. And really what we're doing is the same thing that Mark and I were doing in the last century, right? So this is one of the papers that Mark and I published when we were doing our PhDs, and we were working on bacteria and looking at different solubility systems to extract different subsets of proteins from bacterial samples. And Andrew Gooley here, who a bunch of you will know, I think he was probably Mark's supervisor. He was my co-supervisor, and Andrew and I were co-founders of Proteome Systems with Keith Williams and Mark Wilkins and Nikki Packer. So interestingly, now Andrew is the chief scientist of Trajan, the company that I mentioned earlier, who bought Neoterix towards the end of last year. And so if you take a lot of what we learned 20 years ago and put it into practice here, what you really see, which we expected to see, and that's what we got, is that different washing reagents really affect the subset of proteins that you get out of these devices. And plasma proteins are not just in the plasma. They're bound to a bunch of the cells in blood, and you don't just get all of the plasma proteins out just by centrifuging the sample and taking the plasma off. So if we look at now some applications of the sort of methods that we've developed, this was one of the early things that we did, putting a white blood cell fraction onto these. So Mitra is the trade name that Neoterix use for these little porous plastic devices. So we've put a white blood cell fraction into this, and we've then done a wash, taken it to the mass spectrometer, and we've got 2,100 proteins identified. And when you look at that, there's a really large number, in this case 460 nearly, protein complex genes, and an awful lot of them are membrane protein complexes. In fact, we've got 135 membrane protein complex genes, which, if you go back to conventional proteomics the way we were working on it 20 years ago, getting proteins that were 200 kilodaltons with 24 transmembrane domains was a pipe dream. None of us would ever hope to solubilize or analyze any of the proteins like that. But of course, all we're doing here is trimming peptides off these. We're not really solubilizing them. We're not taking the protein out of the membrane. It's all left behind in the tip. We're just trimming off peptides and taking it to the mass spectrometer. So this method of effectively solidifying blood into these little devices has opened up a whole range of ways of analyzing these samples. And one of the questions that came up really quickly was, would this work with frozen material? So if you had frozen cell pellets where the cells were likely to be lysed, would this work? And we had a whole set of samples. We've got about, this is the same set that I talked about earlier, where we'd done cytokine work. We've got about 40 samples of non-small cell lung cancer, and this was just a subset of them. They're frozen pellets from EDTA anticoagulated blood, where the plasma's been taken off and the pellet's just been frozen. That frozen material goes into these devices just as well as fresh blood. The membranes appear to become stuck inside the tip when you dry it, and then you can wash it. And this was one of the really early experiments we did. So the ID numbers are not particularly high, but these are also quite old samples. And we're about to do this completely again with all of the samples at once. And I suspect we'll see a lot more than 700 proteins in each sample. But this really proved the case to us that you could do frozen material, and we've now done quite a bit more with frozen material, just improving our methods. One of the other projects that has come up that we haven't yet actually started analysing the real samples for is with Helen Wheeler, who's the head at oncology department at Royal North Shore. And Helen has a project looking at cancer patients and their immune response to the COVID vaccines. And what's being taken from these people is we're getting plasma and, again, frozen cell pellets, but they're also using these specific tubes to get PBMCs, and we take a proportion of those PBMCs and put them into these dried blood spot devices. So, of course, they don't look bright red like the picture I've got there, but we've put these PBMC samples in. We've now got over 100 people stored that way. So one of the things we wanted to do was to look at what happens with PBMCs in these tips when you wash them and then trypsinize them. And so this graph here shows you what we're getting. The star there on the left is PBMCs prepped out of Mitra. And so it's taking about 2.5, 3 hours to prep them, but we're getting 3,300 identifications from PBMCs out of these little devices. So 30 microliters of a PBMC sample. And when we compare that to whole blood, so this Venn diagram there on the left, you've got 3,300 unique genes that have come up from PBMCs. Almost 2,000 now we're getting from just whole blood into one of these. And then the overlap in the middle, that 1,400 proteins in the middle, 1,152 of them are in the plasma proteome database. And so that's why I said before, the plasma proteins are not all just in plasma. So when we wash these tips, there's still a large number of plasma proteins left inside the tip, and that's entirely dependent on the stringency that you wash with. Then the other thing that we're looking at, we've come across groups who are working with blood samples that they're taking not to do proteomics, but to do things like circulating tumour DNA. And the thing with those sorts of samples, and this example is with Streck tubes, but there are other ones that do similar things, they have reagents in them that are slow releases of formaldehyde, which ends up cross-linking the sample, and that's where the stabilisation comes from. These samples don't work well at all for immunoassay, so we've tried this and we get quite poor results. But if we put Streck tube purified blood, so centrifuge the sample after collection in a Streck tube, take the plasma off and put the cell pellet into these devices, this is what we're getting. So in this case, 1,700 identifications out of Streck tubes, and again, there's our whole blood with almost 2,000. So once again, the overlap in the middle is really quite large, and there's 1,100 proteins there that are in the plasma proteome database. And so 82% of those things in the overlap are in that database. And so the two stars that I've now added on the left here are the Streck tube and the whole blood data. And the reason I've put that little green box there on the left is that's really the zone that I want to try and stay in for us in terms of sample prep. So look for more IDs, but not in a way where we need to take 10, 20, 30 hours to do it, because it's just not economical from a cost perspective or a time perspective to then do large studies where you need to spend that many hours. We need to keep our sample prep as low as we possibly can in terms of time to be able to execute studies with lots of people in them and longitudinal sorts of studies. And then the last example I'll show you is back to the work we're doing with Mark. So like I said, Mark's the professor of bowel cancer research. He's working with Alexander Engel, who's a surgeon. And when people come into the hospital for colonoscopy, we get blood, they get colon tissue, either polyp or control, taken, and there's a whole nother aspect of the project which is looking at the pathology of that tissue and the genetic modifications in it. But our work with Mark has really been around the blood. The most recent data set we have is almost 50 people. We've put them all, so this is whole blood, into these VAMS devices, and we're now getting about 1,600 per sample. This was the latest data set out of Mark's lab. We've ID'd 2,500 proteins in total. And when you look at the bioinformatics of that, the 100 or so differentially expressed proteins are enabling us to like 80% pull these groups apart. So this is an update on, we've now done about this 2 or 3 different groups of people. The project is ongoing and there's still more people being recruited into it. So this is the kind of thing we're using it for. And I think to close and leave plenty of time for discussion, what I really think now in terms of doing more with less is that these sorts of porous plastic devices really have 2 separate uses. One on the left here is to store aliquots of blood or other samples where you've got fresh or frozen cells in fluid, to collect things that you might have thought were waste fractions that you might have otherwise just thrown away, and store them, and then really to think about them as sample prep devices where that sample becomes solidified and you can work with it in a totally different way to the way we would conventionally work with a liquid sample like blood. And then, of course, there's the way they were intended to be used, which is as sample collection devices to enable us to work with people at home to collect longitudinal samples as a supplement to the kind of thing that happens when people are in the clinic. So that's really the sort of discussion that I had with Jenny and why she thought it would be interesting for me to talk a bit about what we're doing. And then I think there's, of course, a bunch of people to thank. The Sangui Bio team, Liz and Cameron and Rosie, and then the latest person to join has been Natasha, who is our mass spec person. Natasha came from Procan over at Westmead, and I know they were not happy that she wanted to leave and come and join us because she's very good, and that's certainly proved to be the case. She's added a great deal to our capacity, particularly in the mass spec area. Of course, Mark and his group at the Kolling, we work really closely. Mark and I did our PhDs together and have known each other and worked together for a long time. And then at Neoterix, James and Stuart. Stuart's the chief scientist in Los Angeles, and James is really the inventor of their technology, and he's based in England, and we work really closely with them as well. So I'll stop there, and that probably gives us plenty of time in case people have questions.
Thanks, Ben. That was really fascinating. I've got a lot of questions, but I certainly hope that other people do too. So what I might do, I don't think we've got any in the chat, but I think if people would like to ask a question, just pop your hand up. I'll call on you, and I might ask a question to start off. I guess, you know, it's hard to choose, but I guess in terms of your representations, you're talking about getting, you know, quite a lot of representations versus sample prep type, and I can really understand the need to maximize what you're getting versus, you know, the time that you're spending. But I wondered, could you comment on that kind of increased numbers? Are they largely kind of abundant proteins, or are you also starting to dig into the proteome and see things that, you know, tend to be the less abundant proteins? Because I know that's a big issue in sort of biomarker discovery. You know, there's this idea. Oh, well, anyway, I'll let you answer the question. Yeah.
Yeah, it's a good question. I haven't done a really extensive analysis mapping the sort of IDs that we're getting onto an abundance kind of chart of, say, plasma. I think that's something that would be, you know, quite an interesting thing to do. My guess here is that we're still not, in terms of, if you compare this to plasma, you know, we're seeing about 1,000 proteins that are present in the plasma proteome database. So when you look at the best studies that have been done in terms of comprehensive fractionation and then LCMS, you know, they're getting 4, 5, 6,000 proteins. So I don't think that in terms of plasma, what we're getting, we'll be getting into the very low abundance realms of plasma. What I think we're getting in terms of everything else that's in blood are things that are reasonably low abundance, because they're things like mitochondrial membranes and that sort of stuff of white cells, which of course are very low abundance in the context of the whole blood sample. So I think what this is rather than being, we're burrowing into the very low abundance end of things, which I think if you want to get there, you still really need to do massive fractionation or use things like immunoassay, which enable you to get it through a different route. What we're doing is saying there's a lot more in the blood sample that you can get. So you can get intracellular proteins, you can get membrane, you can get both organelle and plasma membrane, and see things in blood that weren't there before. One of the groups we're working with is using the Olink platform, which is an immunoassay system, and what Olink do in-house for their detection. The Olink system, for people that don't know, uses DNA conjugated to antibodies, and there's 2 antibodies for each analyte with complementary DNA strands. So unless you have both of them, you don't get proper detection. And the detection system is either real-time PCR, so we have a system in our lab that does that, and it enables you to work from very small amounts of material, about a microliter or 2. But what Olink do in their own facilities in Uppsala and in Boston is they use next generation sequencing for the detection, and that is enabling them to look from small amounts of serum or plasma at almost 3,000 proteins at once. And so one of the groups that we're talking to has said, look, we do this with plasma. We really are interested in what you're doing with the cellular component of blood. What does the overlap look like? And so when we look at the overlap from either whole blood or things like PBMCs, what we see is that it's 500 or 600 things that overlap. And the interesting thing about that is, of course, probably what the Olink system is seeing are the soluble versions of receptors and things, and what we're seeing are the actual membrane-bound versions of receptors. So I think this is adding value rather than absolutely getting into the extremely low abundance things. That's the long-winded answer.
Yeah, and look, I think, you know, it's a good point. Sometimes in the people sort of, you know, there's sort of a mentality that if it's really difficult and if it's really low abundance, it's somehow more valuable. But of course, that's not true. I mean, you need a biomarker that's easily measured. You need something that's reliable. You know, maybe the important stuff isn't actually down there anyway. Maybe that's just kind of cellular noise or something. So I think sometimes there's kind of the push from technology to do the difficult stuff without really asking the question, is that where the most valuable information lies? You know?
Yeah. I mean, I think getting into low abundant stuff in plasma is certainly interesting. And once you find out what's there, you can use more targeted methods like immunoassay or selected reaction monitoring or something in the mass spec to go looking for it. But I guess our overriding kind of thought here is, what about all of the other things in the blood? So if you're taking a blood sample, don't throw away the cellular component.
Yeah.
And use it and look at that. And what we're trying to develop are methods that enable you to do that in a way that's accessible and not overly difficult or time-consuming or expensive.
Yeah, look, I think it's a very powerful argument. The cells are half the blood, and it could be that some of these molecules are in very low abundance in the plasma because most of them are actually stuck to things on the red blood cells. That would make a lot of intrinsic sense.
Or the white cells or the platelets.
Yeah, so people, please jump in if you've got questions, but I've got a couple of others, but yeah. Now, what else was I going to ask you? Yeah, so this is sort of more of I've got a couple of philosophical questions. So in cancer, I think one of the things that has been a challenge in the cancer biomarker space is that we've already got good biomarkers that are essentially composite biomarkers like tissue grade, stage, and all that sort of stuff. And I think it's often been hard for sort of single biomarkers or even small groups of biomarkers to outperform these other much more complex biomarkers that are, essentially driven by so many different factors. Do you have any comments on that? Is that sort of an inherent challenge for the field? Is that something that we can overcome? What's your view on that?
Yeah, that's
is my question even clear?
Yeah, no, I think it is. Certainly, the groups that we're talking to, they are very interested in blood-based biomarkers, but I would say that a lot of them don't expect that all of the solution to a biomarker issue is going to come out of blood. What they're looking for is potentially imaging biomarkers combined with a blood biomarker. They're interested in things like lifestyle markers. One of those slides I showed where we had the Z-score normalization was simple questionnaire on diet, exercise, drinking, smoking, and stress. And that kind of thing combined with blood biomarkers and potentially in cancers, looking at driver mutations and things like that, it's going to be all of those things combined. It's not going to be necessarily that you'll find 3 markers out of blood that do everything for you. That's really, so it's probably just that I'm agreeing with your position there that, blood's interesting, but that we think there's value boring into it more deeply, but it's not to the exclusion of other sorts of markers as well.
Yeah, that is, I think that's right. It doesn't have to be a sort of an and or, it can be and, it can be this is valuable and this is valuable as well. Yeah. The last thing I might ask is a bit more of a bit of a broader question. You might have already had this. Look, I read the book about Theranos when it first came out, and I guess been very interested in that whole story. I think it's got a lot of lessons for, just quality control in laboratories and research and all sorts of areas. But I did read a quote to say that Elizabeth Holmes had not done any favours for young females in biotech. I'm wondering to what extent that, debacle, I guess, has that had any sort of lasting effects for the microsampling industry, or is that just a sort of a bump in the road that you think you're well over?
Oh, no, it's certainly a spectre that comes up frequently in discussion with, in our world, when you're talking to investors or potential investors, has Theranos and Elizabeth Holmes crawled the pitch for everybody else?
I mean, you really hope not, because there's, that's, yeah.
I think she hasn't really, because what her goal was really to say, we don't need laboratories like Quest or, Lavity or Douglas Hanley Moyer anymore with massive rooms full of fancy instruments. We don't need to go to a phlebotomist and get blood taken. We can put all of that in a box the size of a microwave oven and do it. And I suspect that there are serious companies who are making, and I know there are companies out there making progress towards that, and they will certainly have been affected to a much greater degree by what Theranos did. That sort of thing will come to one degree or another within probably our lifetimes. But what we're doing really is saying there are other ways of using the conventional technology that's here now to look at the sample differently, to just not go, everybody analyzes serum and plasma and therefore we should do that too. We're using mass spectrometers. You know, Natasha runs mass specs over at the CPC at Sydney Uni. It's not run in some magical microwave oven sized device, right? It's a commercially available mass spectrometer. Mark has the same sorts of instruments at the Kolling. The Neoterix devices are commercially available. The kits we use for immunoassay and things are all commercially available. We're really just saying there are other ways, potentially, by deciding that you might not want to read the instructions and do things differently. So I think the Theranos thing was certainly a debacle, but it hasn't had a very big impact on the kind of stuff we do.
Oh, yeah, that's good. Yeah, I just heard the quote about, because she had such a personal profile as well as an individual, that people had felt that, and she was sort of this unique example of a young woman who was in biotech, that the quote was, she hasn't done any favours for women in that industry who probably already, maybe have a tougher time of it as it is. So I just wondered whether that sort of had extended to other, to some of the broader concepts that she was sort of championing, without obviously much data behind it, probably.
I think it's really an example of why, of ultimately how science really does work, and how the network of science and collaboration ultimately weeds that kind of thing out and solves it. Because when people can't replicate something, or they don't go to conferences and talk about what they do and engage with other people, then it ultimately, you figure out that there's a problem.
Yeah. Does anyone have a question that they'd like to ask? Yes, great. Katerina, please go ahead.
Yeah, hi. Hi, Jenny. Hi, Ben. It's Katrina from NSLHD Royal North Shore. We are collecting some samples for the biobank retrospective and prospective. How can we help you with a biospecimen collection? Is there any studies or anything that you need, any future projects that you may need some of our biospecimens, for example? We are collecting healthy controls as well. So,
I mean, that may be the case. I mean, I think at the moment for us, we're not particularly short of samples. So the thing that I was hoping more to get across to people was, when you're collecting samples for your project, maybe thinking about storing some of your samples, or, if you're taking plasma, then the pellet that you've got, rather than throwing that pellet away, if you stored some of that pellet in these kinds of dried blood spot devices, they would be able to be used later as a, rather than just freezing the whole pellet or throwing it away. If you're prepared to, dip half a dozen of these tips in it, then you could store them either at your facility or somewhere like Jenny is running, and they'd be available for other people to look at later. So I think that's the kind of thing that I was attempting to suggest, that there's other ways of thinking about storing your sample, or storing the things that you might consider to be waste.
Yeah. We were originally considering collecting or freezing red blood cells. However, I mean, it would be good to do a little trial and use some of your kits and just store at the biobank if you would like. If you find, maybe we can talk with Bernie maybe later on at another stage. If you need any help, we're happy to help. Yeah.
Sure. Yep. We can have a chat when Bernie gets back from wherever he is around the world at the moment.
He is back, but he's got COVID, unfortunately, so inevitable. So, but he's back. Yeah. Hopefully he's okay.
Yeah. Okay.
Thank you.
So Ben, do you have a link that you could put into the chat where if people want to obtain the devices, how they could do that?
Sure. What I need to be
I don't want to put you in the spot, but or you can send it through to us later. Hang on, here we are.
It might be easier for me to send it through to you later if that's possible.
That's great. That'll be fine. Yeah, look, we can do that.
I don't even have
send
my
that
web
out.
browser open.
No, that's good. That's a good way of not being distracted.
Yeah. I'll email it to you and Michael later, and you can potentially put it up there.
That'll be good. Yeah, we'd really like to do that. Yeah.
Okay.
Look, if there aren't any other questions, I think we can maybe join in thanking Ben for a really interesting talk. I've just found that fascinating. I loved it from the minute where you showed that graph where you showed all of the signal in the red blood cells and nothing in the plasma. I think that's the kind of data where really exciting discoveries are made. So thanks very much for sharing all of that with us, and thanks to everybody for making the time to join us, and we'll see you at a seminar again soon. Thanks everyone.